So I'd like to present our research deciphering the genomic landscape of signal-based traits through latent space analysis. This research was done by myself, Natan Lubman, and Dr. Nadav Rappaport. We'll talk shortly about the prevalence of cardiovascular disease. We can see that it's one of the most, if not the most prevalent disease that causes deaths around the world, and we'll give it a lot of importance here in our research. We'll start by talking about general stationary stationary genetic traits. So when we're looking at genetics, a lot of time we normally talk about our, let's say, eye color. It's something that we're born with and we stay with mostly our whole life, and it doesn't really change like other traits. If we're talking about hair color for most of our life, it does change at some points, but it's not something that we can frequently see different changes and repetitive behaviors throughout time. So we'll talk about a signal that's a bit less stationary, a bit, but it's not stationary. We'll talk about our pulse rate, but this pulse rate is through a what you'd call a PPG device. That's an optical device that can measure the change in volume in the blood in your vessels, and what we actually see is that it has different behaviors when it reaches different points in our vessels, blood vessels, arteries. The deeper it reaches, the more reflective waves we can see. Eventually, we'll see a single wave, but this wave is built by multiple different waves. So until now, those features have been handcraftedly extracted, and we want to show a different method that might find different features that we can't see with our naked eye and might have very strong meaning when we're talking about the connection to genetics, and might be able to show genetic drifts throughout our lifetime, because this is a changing signal. We're looking at the PPG as a generic signal, but we can also look at other signals like ECG, which has clear physiological explanations, but we might not be looking at the signals or that area of its behavior properly when we're talking about genetics. We can see that ECG itself is also built, it's also a superposition of multiple signals inside the heart, and it's not just a single signal, which makes it interesting. If it was a single signal, it might not have as much, this method might not have as much effect. This is just a general overview of the PPG technology. We can see that there's a reflective and transmissive method, which each of them checks the volume in the blood vessels when light goes through. So the data set itself was taken from the UK Biobank. There's over 500,000 subjects with about 170,000 signals, useful signals of PPG. The signals went through all sorts of processing procedures, normalization, length adjustments, noise was removed, and it was upsampled to a higher frequency, which showed the data or showed the information a bit better. What we suggest is a CNN autoencoder model, where we insert a signal, which is their olive length or normalized to the length of 400. That's the default inside the data set. The default is 100. We upsampled it to 400 without losing the behavior of the signal itself. And we inserted into a CNN autoencoder, which different filters were able to extract different types of data. And at the end, we got a latent space of 40 features, which was an optimization that we did throughout the procedure of building this autoencoder, where eventually we're looking at extracting or reproducing the signal that we have. Eventually, these 40 features, they're very important because they are stationary, which means for every signal, that means unlike a time-dependent signal, where if you change 2 points in time, it loses its sense and meaning. If you change the location of these features in time, it doesn't have that much importance, because these features already extracted in some way behaviors inside the signal which represent the time itself. So suddenly we have features which we can compare to genetic data, where here the peak can appear in different locations, and we can simply create a type of embedding that we can use later on. So we have to remember these 40 features of the latent space of this model. So the whole procedure itself is we take data from the biobank, we do a signal processing stage, we take the latent space model, and with the genomic data that we also took from the biobank, we inserted into a GWAS analysis. This GWAS analysis, after we finished it, we got SNPs with p-values. We filtered the most significant values, and we filtered them. We inserted them into a haploreg, and we sent them into a clustering model of string, which has a pre-processing knowledge, and we got gene clusters, which later on we can create from them phenotypes and phenotypic predictions and risk factor models. So we found hundreds, 500 and over 500, almost 550 SNPs associated with the signal itself. 26 were already found on GWAS, including electrocardiography, pulse pressure measurements, atrial fibrillation, skin pigmentation, QT interval, and all sorts of other things that are cardiovascular connections. We can see a strong cardiovascular connection here, and here a regulatory connection, which have a lot of meaning after we go through the data, and that's something you'd be able to look into when we publish the research in a more organized way. We can see that it has a lot of has a very strong connection to sudden death, heart block, supraventricular arrhythmia that might be able to help us later on give a prediction or a risk factor for this information. We can also see that in the pathways that these genes are very central or in a very close area together. Again, this is the regulatory cluster, which is very interconnected, and we can see a cluster that is potassium channels, which is also very connected to cardiovascular information. And we can see that here too, the genes, the ones that are signed in red, or the ones that are in the cluster, are very interconnected here in the KIG pathway. So discussion, it seems like there are hundreds of SNPs of importance when observing. Many of them are associated with encoding gene areas. Some SNPs have high p-values using multiple features, which shows maybe their importance and maybe interconnection between the features themselves. There's proximity of the SNP's location, which can show stronger confidence. Engineered features seem to be able to recover many more genes of importance compared to the use of simple features, indicating that there are probably hidden representations which we can't identify with our naked eye. So we might be able to later on take this and make a real-life implementation and insert it into watches or oximeters. We can also use different types of signals, like respiratory rate signals, ECGs. This is the PPG that we showed earlier. And the conclusion is that there's important information regarding the user's genetics in the data. We might be able to use data to recover features, to identify some of the user's genetic traits. Using a user's latent vector, we might be able to identify the user's drift on the feature space over time and see deterioration or improvements in their physical condition. And we might be able to create a new risk factor using this information. That's it. Thank you for listening.